last speaker of the day is Professor Tal Pupko from the Faculty of Life Sciences. לא עובד?
זה עובד, צריך להכניס את כן. מעולה, יש לך גם מיקרופון.
I think I don't need the microphone. I will speak like Now it's okay? Okay. Hi, so thanks everyone for staying. I'll try to be brief. Okay, so I'll talk about, I won't talk about COVID, and I'll not talk about viruses actually. I will talk about bacteria, pathogenic bacteria. And what you see here is a syringe. It's a type 3 secretion system. This is the, let's see if I have a pointer here. Yes. This is the bacteria, gram-negative bacteria have two membranes, the inner membrane and the outer membrane. And this syringe is going from the bacteria membranes all the way to the host. And what these protein complexes do is they take proteins that are bacterial protein all the way through the needle, injected into the host cytoplasm, and then in the host cytoplasm they do a lot of things to the host, like inhibit apoptosis, take control of the cell to the benefit of the bacteria. Okay, so my goal in my research is to understand how pathogenic bacteria actually do what they do. And one of the first goals in order to understand the pathogenicity at the molecular level is to identify the effectors, which genes encode effectors that are secreted by the type 3 secretion system. And this is important for many diseases, and the type 3 secretion system is used by a lot of important pathogenic bacteria, including Yersinia, that is responsible for the plague, Shigella, pathogenic E. coli, and Xanthomonas. Xanthomonas is interesting because it's pathogen on human, but also in various plants. And there is also Xanthomonas, which cause disease in various plants, including tomato, apricot, lemons, rice, and others. Okay, so how can we identify the genes that are encoding for effector proteins? Basically, we have a test. We can, if we have a putative effector, we can clone it, fuse it with the Psi-A toxin, and then if it's secreted through the type 3 secretion system into the host cytoplasm, the Psi-A toxin will interact with calmodulin. Together they will form an active complex that will catalyze the transformation of ATP to cyclic AMP. And we have a kit to sample cyclic AMP levels. So this is when we don't have the type 3 secretion system, very basal level of cyclic AMP. If we secrete a putative effector, that is a genuine effector, we have high level of cyclic AMP. And if we use an empty vector, it's not an effector. So in theory, we could take all the 4,000 genes, clone each one of them, and test if they encode effectors or not. But this is not practical. So before in my lab, we developed computational tools to identify type 4 secretion system effector. It's the same concept. And we work closely with Professor Gil Segal and Dr. Dudu Burstein. And we developed computational tools to predict the effectors. And the idea in this work and in the work that I show you is to look at features that can tell us hints about whether or not a given protein is an effector or not. So, for example, one thing that actually Gil noticed in type 4, but it's also true for type 3 effector, is that the effectors, many of them share common domain. Not all the effectors have this domain, but if you have domains that are shared between known effector, it's more likely that you are an effector, that the protein is an effector itself. Also, the effectors tend to reside in genomic islands in the bacteria. They are not equally distributed. So if a gene is next or in the proximity to known effector, it's more likely that it encodes an effector as well. And also the GC content. Many of these pathogenicity effectors are transferred by lateral gene transfer. And when a gene is laterally transferred, often the GC content of the gene is different from the GC content of the recipient genome. And also, we took every hint we could to detect effectors. For example, if you see that the gene is as a homologue in a bacteria that does not encode type 3, then probably it's not an effector. And also, of course, we look for similarity against effectors, and also we blasted each of the bacterial genomes against the host genomes, like human. And because many times effectors, since they have to interact with human proteins or with eukaryotic proteins, they have domains that are found usually in eukaryotes. So we took all, and finally, sorry, also, I'm jumping. We also have the regulation, okay, sorry, it disappeared. But we know that probably effectors are co-regulated, so we can probably also take this into account. And secretion signal and many other features. And the idea is to combine everything in machine learning. So I won't actually give, I won't explain the basics of the machine learning concept, but the idea is if you just have two features, let's say GC content and the hydrophobicity of the N-terminal, the secretion signal is probably reside in the N-terminus of proteins. So you will have, each dot is a protein. So for example, this in red we see proteins that are not effectors. So they have low GC content and low level of hydrophobicity. And effectors, they are here. So if we don't know what this gene is, probably it's an effector. And this gene that we don't know, probably it's not an effector. So this is a very simplified version of reality. In reality, we don't have two-dimensional. We have over 100 dimensions. We don't find a line. We find in hyperspace, and the separation is not as good. But this is the concept. So we apply the host. Now it's back. We applied many machine learning algorithms and selected the best, the classification algorithm. So this is an overview of what we did. We have the machine learning algorithm. We have few known effectors. We extract features from the known effector. We extract features from the non-effectors. We ask the machine learning to learn what differentiates effector from non-effectors. And then we put all the rest of the genes and ask the machine learning to predict which genes are effectors and which are not. And then we validate them experimentally and we reiterate the process. So it worked. The nice thing is that we were able to discover the effectors in various pathogens. For example, in Acidovorax, it's a pathogen of melons and watermelons. We were able to experimentally identify, using the machine learning approach, 7 new effectors. Basically, we tested all the 7 high predicted of our top scoring predictions were true, and we identified novel effectors, and later their function in the host cell, we started to study it. And interestingly enough, in this case, the most informative feature was that we found out that the genes are only there. They don't have homologs in closely related bacteria without a type 3 secretion system, and also a regulation motif in their promoter. Also, with Ehud Banin from Bar Ilan University, we discovered the novel effector in Xanthomonas aeruginosa, a human pathogen that is common in cystic fibrosis patients. This was very challenging because previously there only 4 effectors were known, and we increased the number to 6. And also with Professor Guido from our faculty, and Doron Tepper, who is now a faculty at Vulcani, we identify effectors in Xanthomonas bacteria that infect tomato. And also Professor Yitzhak Barash, also from the plant science school, he came to us and he said that there is a pathogen, Pantoea agglomerans, that affect Gypsophila, these white flowers that we often see in wedding. And he asked us to find effectors in the bacteria. So we said, okay, give us the genome and let's see. And actually the genome was not sequenced. So we sequenced the genome, predicted effector, evaluated them experimentally, and these were the first effectors that were identified in the Pantoea that infects this flower. And recently, also we collaborated with Professor Gad Frankel and found a novel effectors in Citrobacter rodentium. This is a project of a PhD student from my lab, Naama Wagner. And there is I'll show you.
Around us, there are different microorganisms that can damage our health, whose mechanisms of infection can be sophisticated. Among them, some bacteria target our gut and make us sick. These disease-causing bacteria attach to our intestinal cells. They use a syringe to inject harmful molecules called effectors, which work together to take control of our cells as a hacker seizes control of a company's software. The bacteria use these effectors to evade our defenses, manipulate cells, and colonize the intestine. Antibiotics are not often effective against these infections. A deeper understanding of this cellular hijack phenomenon would help the development of alternative treatments. However, the involved cellular pathways are so huge and complex that scientists still don't know how the effectors cooperate to hack our cells. That's why a team of researchers from the UK, Spain, and Israel have joined forces to tackle this difficult challenge, combining lab experiments and artificial intelligence tools. They have used a mouse-infecting bacterial species to study this process in vivo, aiming to better understand how groups of effectors collaborate during the infection. Researchers generated over 100 bacterial strains with tailored sets of effectors and investigated their capability to colonize.
So we did not but we predicted the effectors and they were validated, and we think that now we have the full repertoire of effectors in these pathogens. And since we repeated this work quite many times, we developed a web server that you can just, it's the only available web server for predicting factory effectors, that you can upload the genome and you get a full list of all the effectors in the genome. So you don't need that anymore. You just only plug in the genome to the web server and get the prediction for the type effectors. And actually what we
we do now is we study disease of Xanthomonas that infect strawberries, and it's a real problem in agriculture around the world, and we successfully predicted new effector and validated them experimentally. This is again a collaboration with the rodentium from BIOINFORMATICS. And these are the effectors that we found. And now we are working on also bacteria that infect geraniums. And we took field isolates, we sequenced the genome. We are now in the process of cloning the genes and validating our predictions. And we are also interested in the level of evolution. So we try to understand the evolution of type 3 secretion in systems and effector in various Xanthomonas species. Now Xanthomonas, there are some environmental Xanthomonas that can be found in rainwater, and today, together with our collaborators in Alabama, we are trying to use phylogenetic methods, tools to predict effectors, to understand the evolution of the system, and over 1,000 genomes of Xanthomonas. So I just want to acknowledge the many people that did the work. Now, in the PhD student in my lab is doing now the work, and she was a from the science center. David Pupko was a faculty member, but he started this project when he was a PhD student at my lab. And he said that introduced me to the field of effectors, type 4 effectors, and a lot of my knowledge, and Guido and Barash and Banin and that friend from the UCL and Spanish. Thank you.
After you find the pathogen, what do they do with this information?
You ask what's the next step after I find the effectors. So the difficult part is to understand which eukaryotic proteins interact with each effector, or sometimes effectors that interact with other effectors, and try to find the network. So Professor Gil Segal said that each effector is a PhD student, but it's I cannot contribute much to this effort computationally. So it's you have to do you have to study protein-protein interactions. You have to overexpress it. It's been a lot of experimental work. At least the identification of effectors, I could really use machine learning to do it efficiently. Any other questions? Thanks everyone for staying.